We're in the digestive system. Where does food enter the body? The oral cavity, that's right. And from there, it goes where? All right, technically into the oropharynx, good. And then into what region? The laryngopharynx, good. And then from there, where does it go? To the esophagus. And then to the stomach. And from there, the small intestine, specifically the duodenum, which is followed by the jejunum, followed by the ileum. Good. And then from there, the large intestine. What part does it hit first there? Cecum, followed by ascending colon, followed by transverse colon, followed by descending and sigmoid colon. Then we have the rectum and anus, right? Last time we also talked about the fact that there were 6 processes that were going to help us with digestion. Remind me, what are those again? We have ingestion, good. What else? What? Propulsion, good. Mechanical digestion and good. So mechanical and chemical digestion. What else? Absorption. And the last one? Defecation. Where does ingestion happen? Oral cavity, that's it, right? What's the only other one that only happens in one place? Defecation, because that happens with the anus, right? As we remove the stuff that the body couldn't use. Where does propulsion happen? Throughout which part of the digestive system? Remember, we divided it into 2 major parts. Only along the alimentary canal, because the other part was classified as what? The accessory. And remember, the accessory organs never actually held the food stuff, so there's no way that they could really help with propulsion, right? As we move this stuff through the GI tract. Where did mechanical and chemical digestion happen? Because for the most part, they happen in the same places. Small intestine for sure, and that actually does the most chemical digestion. What else? Stomach for sure as well. What else? The oral cavity, where it all starts out. Absorption, where does most of that happen? Small intestine. What else helps us with absorption? The stomach does, and large intestine, good. Although those areas are going to be more responsible for absorbing things like water and electrolytes and drug metabolites, those kinds of things. Whereas the small intestine, we say it does the most absorption because that's where most of our nutrient absorption is actually happening. So our goal for today is to get through the first half, essentially, of the alimentary canal. So we're going all the way from the oral cavity to the stomach. On our way, we're going to be talking about deglutition, which is our fancy term for swallowing, and we'll be looking at how some regulation happens in these organs. So we better get moving. You guys already know a little bit about the oral cavity because you started studying it in lab last week. You want to make a note that an alternative term for the oral cavity is the buccal cavity. So if you see B-U-C-C-A in a term, that's telling you that that term is related to the oral cavity. And you know a little bit about it already because we introduced parts last time, and we talked about some things here when we talked about the respiratory system. So what kind of epithelium would we have lining the oral cavity? Stratified squamous. That's exactly right. Why? What can't get through? It's going to make it harder for what? Why do we need a thicker wall here, guys? To protect from what?
Bacteria.
Yeah, things like microbes, right? The mouth is sitting right here next to the external environment, which we know is filled with all kinds of nasty stuff that we don't want to get into the body. So it makes sense that we need a thicker wall. Also, I heard some people saying something about the food. It's true, the food is in a more solid form at this point. And so it can create more friction where it could damage some of the cells. Makes sense we'd want some as a backup, right? So that the wall isn't breached. We need more layers here to protect from any friction damage. One thing that we didn't learn about in lab that I want you to make a note about for lecture is the oral orifice. So that's technically the space between the lips where the food is actually going to enter the body. So from the oral orifice, it's going to go into that space between the lips and the teeth. What was that space called again? Vestibule. The vestibule, good. And then into the main lumen of the oral cavity before it hits, what was that back opening? The fauces, good. And then it gets into the oropharynx, right? So when we think about what's making up the sides of the oral cavity, we clearly have the lips at the front, at the anterior side. We've got the palate at the top, the superior side. You know about the hard palate and the soft palate a little bit from lab. What hangs on the back of the soft palate? What's that back part? Uvula. The uvula. Why is that going to be important?
Blocks large things from.
Yeah, it's going to close off what? The nasopharynx and nasal cavity. This is something we've talked about before. We're just going to hit that point briefly today since you already know a little bit about that. Clearly we have the tongue at the base and we've got the cheeks at the sides. I want you to know a little more detail about the hard palate for lecture. So you do need to know that the maxilla makes up the anterior portion of the hard palate. Remember from A&P 1, it's technically the palatine processes of the maxilla. And they're going to meet up with the palatine bones on the posterior side of the hard palate. If you run your tongue along the top of along the roof of your mouth by the hard palate, if you go from the front to the back, you'll notice that along the front side, it feels like there are ridges. That's because of the way that the hard palate is structured, and that's important because that's going to help us to increase friction in the oral cavity. So that's going to help us with some of that mechanical digestion, kind of mashing things up a little bit as we're moving food around in the oral cavity. The soft palate, you need to know, is made of skeletal muscle. That includes the uvula. And yeah, we already talked about why that's important, because the uvula, when we swallow, is going to swing kind of backward and up a little bit to close off the nasopharynx and nasal cavity. So that way when we're swallowing, the food doesn't go up there. Of course, that's not a perfect system, right? Because I'm sure everybody has seen somebody laughing while they're trying to drink a soda or something, and then the soda goes out their nose. Well, that's because they were laughing and the uvula didn't close properly when they tried to swallow that soda. The tongue, it helps us with a lot of functions here in the oral cavity. So you do need to know that, since it's this muscle that's sitting at the base of the oral cavity, it's going to be really important for moving the food around, making sure that we get the food by the teeth so we can mash it up when we chew. You also need to know that it's going to help to mix up the food stuff with the saliva. And so that's one of the tricky things about the digestive system. After the food enters the body, we don't call it food anymore, because it's not just food. We mix it up with other stuff. So when the food stuff is ready to leave the oral cavity, we change its name. We call it the bolus. And it's going to be the bolus that moves through the pharynx and the esophagus to go down into the stomach. Then we change its name again because we're going to be mixing it up with other stuff. So make a big star by that. That's an important point. You need to know what we call the food in the different parts of the alimentary canal. Clearly, the tongue helps us with other things that aren't necessarily just digestive things, right? It helps us with speech. We move it around a lot when we talk. It has taste buds, which you studied in A&P 1, so you know that it gives us our sense of taste. It also is going to help us with swallowing or deglutition, because what's going to happen is it hits the front of the hard palate and kind of squeezes the bolus back toward the pharynx when we swallow. When you studied the tongue in lab, a lot of people pointed out that, wow, there's a lot more muscle here than I expected. Well, it's because there are intrinsic and extrinsic muscles that are associated with the tongue. The intrinsic ones help it to change its shape. So if you can roll your tongue, it's those intrinsic muscles that allow you to do that. The extrinsic muscles reposition it. So those would be the muscles that, for example, allow you to stick out your tongue. One last note about the tongue here. I do want you to know that the mucosa associated with it is modified by the base. It forms a structure called the lingual frenulum, which is essentially a structure that acts like an anchor on the front side of the tongue. All right, salivary glands are going to be important. What you don't know from lab, though, is that we have extrinsic and intrinsic salivary glands. In lab, you studied the extrinsic ones, the ones that are found outside of the oral cavity. But we also have intrinsic ones. The extrinsic produce the most saliva, and so they're going to be more important when we actually have food in the oral cavity. The intrinsic ones are more important for just keeping the mouth moist. And sometimes you will hear them referred to as buccal salivary glands because they are actually in the oral cavity. I prefer intrinsic and extrinsic because they tell me specifically where they are. So, what's the saliva going to do for us? For one thing, it's going to cleanse the mouth. I already said it's going to help with moistening things. Part of what it's going to do is moisten and dissolve the food, which we know because we studied the enzymes that we find here. We said that we mix it up with the food to make the bolus, the stuff that's going to leave the oral cavity. And I mentioned enzyme. What is the enzyme that my slide is referring to?
Salivary amylase.
Salivary amylase. Very good. And that's the most important one here, right? But we've made a little note that there was another enzyme. What was that other enzyme? In the oral cavity? What's the other one in the oral cavity? Lingual lipase. Very good. Very good. Remember, lingua means tongue, so that's helping you to see that this enzyme is in the oral cavity. And lipase, what do those break down? Fats or lipids. Why did we say that was a more minor enzyme here? Why does it not really help that much with lipid digestion? It's true. It hasn't been exposed to bile salts yet, and that means that the enzyme can't get to the middle of the fat bubble yet, right, or globule. So what was that process that needed to happen before we could really break down the lipids? Emulsification, exactly. Because emulsification hasn't happened yet, lingual lipase is very limited in its ability to chemically digest fats here. That's why we focused more on salivary amylase, because carbohydrates are hydrophilic, just like our enzymes, which means that it's easier for the enzymes to act on them. All right, so we know what the saliva does. We need to look at its parts. The parts actually have a lot in common with the sweat. So the sweat was mostly water, just like any other body fluid. No, saliva is too. The sweat was slightly acidic. Saliva is too. Why would it be beneficial for these secretions toward the outside of the body to be acidic? Exactly. That's an antimicrobial feature. If we lower the pH just a bit, if we make it acidic, it's going to be harder for bacteria to live there. Just like all body fluids, we have electrolytes or ions here as well. We've seen metabolic wastes and a lot of these secretions that are going toward the outside of the body. And you guys know about the enzymes because we just talked about them. From general biology, you should have learned that every enzyme has an optimal pH at which it works. So since the secretions here are slightly acidic, it makes sense that the salivary amylase and lingual lipase will work best at a pH that's slightly acidic. We also have a lubricant that's part of the saliva, mucin, which is going to be important for reducing friction, especially as we start to swallow. Don't worry, the building's not falling apart. I'm sure, I promise. And we have antimicrobial chemicals. So we've seen defensins and lysozyme before. And it makes sense that we would have these as part of the saliva because this is a great entry port for bacteria. And this is why they tell you, when you sleep, it's best to try to breathe through your nose because if you breathe through your mouth, what happens? It dries out, right? Your mouth dries out. And if your mouth dries out, then we don't have all of these chemicals to help fight the bacteria that want to grow in our mouths. So, thinking about what the salivary glands do. You know about the saliva, you know about its function. The intrinsic salivary glands are most important for just helping to keep the mouth moist, in part to help keep the bacteria out of there. The extrinsic salivary glands, because they make more saliva, they're going to be most important when we have food in the oral cavity. And that makes sense, right? Because if we have food, we need to have plenty of chemicals to help with breaking down that food and lubricating that food before we get it out of the oral cavity. Why do I have the word parasympathetic underlined? Yeah, because that's the end that's going to speed up these salivary glands. In other words, that's the end of the autonomic nervous system that causes them to secrete. Sympathetic does the opposite, right? Mm-hmm. So if the sympathetic nervous system is acting at too high of a rate, like if somebody is a really nervous speaker, you can always tell who your nervous speakers are because they always have a bottle of water with them when they give a talk. Why? Because sympathetic nervous system's kicked on, their mouth is getting dry, and that makes it hard to talk. I do want you to know the name, the technical name for dry mouth. That's xerostomia, which I have on this slide for you. All right, from the oral cavity, we know the food is going to hit the pharynx next, except we don't call it food anymore. What is it? The bolus, that's right. What regions again? Oropharynx and then laryngopharynx, good. What kind of epithelium will we find in these portions of the pharynx? Stratified squamous still, because the bolus is still capable of causing some friction here. And do remind yourselves that we don't just have food that moves through these regions of the pharynx, we also have air that can move through there as well. Otherwise it would never make it to the larynx. right. Esophagus is next. So we know that this is connecting laryngopharynx to the stomach. One thing that's tricky about the esophagus is it's such a long tube that we see changes in its wall layers. So we talked about the layers of the wall last time. Remember, mucosa sat next to the lumen. It had 3 sublayers. Then sitting outside of that, we had what? The submucosa, which was made of connective tissue, and we were looking for blood vessels there, right? And then sitting outside of that, we had muscularis externa. Muscularis mucosa was one of the sublayers in the mucosa. That's why mucosa is in the name. So muscularis externa sits outside of that. It usually has 2 sublayers, right? And then outside of that, we either have serosa or adventitia. Okay, so if we look at this picture from the book, it's showing us that the mucosa has stratified squamous epithelium. And that is true at the upper portions of the esophagus. But everything in the body changes little by little. It's not like you're going to have stratified squamous just automatically, bam, there's a line, and then it turns into another type of epithelium. So this is going to slowly be altered and modified so that it becomes simple columnar epithelium before we hit the stomach. And that's because that's the type of epithelium that we have in the stomach. So make a note about that. It's not always stratified squamous. Before we hit the stomach, that epithelium changes. Another thing that we're going to see changing is the submucosa is going to have different types of glands here in the esophagus. It has mucus glands, and we know what mucus does. What's mucus going to do here? Lubricate, exactly. Exactly. That muscularis externa is going to change as well. At the upper portions of the esophagus, it's skeletal muscle. That's why if you swallow, you can feel contractions through your throat. But you stop feeling the contractions past that because smooth muscle takes over and you don't have to think about that contracting. We also need to make a note that the outermost layer is adventitia here. And that's something that we made a big point about last time. Do you guys remember what the difference was between adventitia and serosa?
It's just the location.
It's not just the location. Location is a factor because if we think about the esophagus, it's not always found in a body cavity. The tissues are different. They are different. With adventitia, we only see connective tissue. With serosa, remember, we had connective tissue and epithelial tissue. And they had different functions. What do serous membranes always do? Serous membranes have that cavity filled with fluid that acts as a lubricant. So serous membranes reduce friction, especially in body cavities where we have a lot of organs moving next to each other where they could rub up against each other and cause friction if we didn't have those membranes, right? So adventitia, since it's just connective tissue, and it can be found in areas where we don't have body cavities, that acts more like a glue. It helps to just glue the esophagus onto surrounding tissues like in the neck. So, if we look at the processes of digestion that happen in these 3 parts, it's easiest to do all 3 of these guys together because they have related functions. We know we have ingestion happening in the oral cavity. We also know that we have mechanical and chemical digestion, mostly also in the oral cavity. Mastication, that word on my slide under mechanical digestion, that's our fancy term for chewing. So mastication is chewing, which is mostly voluntary, but you know, like if you're chewing gum, you're not always thinking about chewing the gum. Your mouth just kind of does it. So it is partially reflexive. And we have propulsion, which we know is happening through all three of the parts. So we need to look at that swallowing or deglutition in a little more detail before we leave this part of the alimentary canal. It's going to happen in two major phases. We have the buccal phase, which obviously is going to happen in the mouth. Exactly. This is voluntary because we have skeletal muscle working, like with the tongue and in some of our upper tubing there. Pharyngeal esophageal phase is going to be involuntary. And I know we have some skeletal muscle in there, but remember, a skeletal muscle can be controlled in a reflexive way. So let's take a look at this. This is a figure from our book. And we can see it's telling us that we're about to begin the buccal phase. It hasn't actually started yet. So we have the bolus just kind of sitting in the oral cavity right now. Notice that right now the uvula is sitting right where it has been in all of our models and pictures so far. Also, when we go to swallow this food, what else is going to need to move? The epiglottis is going to need to move because it's going to need to swing down and cover up the larynx, right? And what else are they showing us on this figure? We've got a sphincter. We've got an upper esophageal sphincter that right now is contracted and closed. So when we swallow this bolus, when deglutition begins, that sphincter is going to have to relax and open up to allow the food to pass down into the esophagus. So when we see the buccal phase begin, notice what happened. The tongue moved upward to push the bolus toward the back, right, toward the fauces and the oropharynx. As it did that, the uvula closed off the nasopharynx just like we said it would. Also, notice the epiglottis has bent down to cover up the larynx, also just like we said it would. And our sphincter, even though they're not showing us it now, it's clearly open because we can see a lumen in that tube now, right? Once the bolus moves past the sphincter is going to close back up. Why does that make sense? What? Think about it. We have contractions happening in this tube, right? Muscle contraction that's helping us squeeze the bolus through it, just like squeezing that tube of toothpaste example. So what would happen if we squeezed our tube right here? Would all of the stuff just go down? Yeah, as we squeeze it, some might try to go back, right? The other way. So we've got to close off that sphincter to make sure that everything keeps moving forward so that we have true peristalsis happening here, where we're moving the food stuff toward the opposite end of the tube. So with the pharyngeal esophageal phase, we can see smooth muscle is continuing that peristalsis, moving the bolus toward the stomach. However, before we hit the stomach, notice what we've got, another sphincter. So you need to know about the gastroesophageal sphincter, and that's a pretty important one. Why is that one really important? Yeah, because what's the environment in the stomach like? It's acidic, and it has protein digesting enzymes, right? It's a really harsh environment. We don't want to expose the esophagus to that. So that sphincter only opens when the bolus is moving into the stomach. Then it's got to close right back up to protect the esophagus from those secretions. If that sphincter isn't working properly, well, that's what causes heartburn or gastric reflux, where the acidity is coming up the esophagus, and the esophagus doesn't really have a way to protect itself from that. So it gets damaged from that, and if it happens too much, scar tissues can build up in there, and people sometimes need surgery when they get older because of that. All right. In this last figure, we can see the gastroesophageal sphincter has opened, and now the bolus is entering the stomach. We know that sphincter will close after the bolus is fully in the stomach. So it's time to talk about the stomach. We see our 4 major layers of the wall for the stomach. Notice what they're showing us here. They've taken just a little chunk out of the stomach and they blew it up to a bigger size so we can look at it. So we have the lumen of the stomach up here, and then we can see our layers. One of our major layers has more sublayers than it should, at least according to what we've learned so far. Which major layer has more sublayers? The muscularis externa has an extra sublayer there. It's got an oblique layer, not just circular and longitudinal. This is significant. You care about that oblique layer, and the reason why is that is the layer that does the strong contractions that help the stomach with mechanical digestion. So make a big note about that. That is really significant. It's supposed to be there. But this is the only organ that has it because it helps with mechanical digestion here. Now, if we take a closer look at the mucosa, we're going to see some things here that we haven't seen in the other organs. So notice that the mucosa is mostly made up of gastric glands, and those are connected to the lumen by these gastric pits. So the gastric pits are kind of like the tubing that's connecting the gastric glands to the lumen of the stomach. And they're highlighting that simple columnar epithelium that we know there's going to be lining the stomach. So notice it lines the lumen as well as the gastric pits. When we get into the gastric glands, we start to see modifications to those cells. So we're going to be changing their names based off of what they secrete in the glands. So let's take a closer look at those. There are going to be 4 different types that we're going to have to know. We have mucous neck cells, parietal cells, chief cells, and enteroendocrine cells. If we look at the gastric gland, notice what are the majority of the cells? The chief cells, right? They're the purple ones. It's mostly purple. We see a few parietal cells and enteroendocrine cells here in green. They're kind of interspersed between the chief cells. Mucous neck cells are found closer to the top, closer to those gastric pits. The reason why we have so many chief cells is because those are the ones that make our enzyme. But notice, when they secrete the enzyme, which, by the way, what was the name of that enzyme in the stomach? Pepsin, exactly. Notice, when they secrete pepsin, it's not pepsin. It's inactive. So its inactive form has a different name. It's called pepsinogen. And that's a common trend that we see with all protein digesting enzymes. When they're secreted, they're originally secreted in an inactive form. And that's kind of the body's way of protecting itself. Think about it. We know cells have a lot of protein, right? So it doesn't make sense for a protein digesting enzyme to be active inside the cells of the body. Otherwise, it could damage those cells, could kill them. So in order to activate pepsinogen and make it pepsin, there are a couple different ways we can do this. If we look at this little box here, it's showing us how this happens. So notice hydrochloric acid is going to be made by the parietal cells, and that acid is actually going to convert pepsinogen to pepsin. That's why it's sitting by the main arrow. But what else is that little box trying to show us? Why is there that other arrow? It's not showing us that it's reversible. If it was, it would just be a flat, straight arrow, right?
It's showing that positive feedback can create more.
Ah, yeah. It's showing us that as we make more pepsin, it can go back and cause more pepsinogen to be converted to pepsin. And you're right, that's positive feedback, and that's important. Remember, guys, we hardly ever see positive feedback, right? So anytime we do, that's something that's a major point. So part of pepsin production is positive feedback as pepsin converts more pepsinogen to the active form. Parietal cells secrete more than just hydrochloric acid. They also secrete intrinsic factor. Hydrochloric acid is going to make the secretions somewhere around pH of 2 or so. So from 1.5 to 3.5, pretty acidic pH. And that's important for a number of reasons. One, what we just talked about, that helps to make pepsin. Two, it's protective. It's going to kill any bacteria that get into the stomach. In fact, when a person has food poisoning, that's not usually because of live bacteria. That's because the bacteria got into the stomach, and then as they were killed, they secreted toxic substances that then got into the body. So it's actually the chemicals from the bacteria. Also, we know what acidity or changes in pH can do to proteins. They can denature proteins. What does that mean if a protein gets denatured?
It dies.
It loses its 3D shape, which in the body means it can't act, right? Mm-hmm. But with our food stuff, this is an important thing because it's kind of like we're unraveling the protein. We haven't broken any bonds, but we're changing its shape, which is going to make it easier for pepsin to get in there to start breaking some of those bonds, some of those peptide bonds. That way, we're making our big protein into smaller polypeptide chains, right? Which we'll break down more later. I want you to make a brief note about intrinsic factor. This isn't something that helps us right now, but it will help us when we get to the small intestine. It's going to make it easier to absorb vitamin B12. Enteroendocrine cells, that name gives you a hint about what they do. What does endocrine mean? Endocrine, like endocrine system, makes hormones. You're exactly right. So these enteroendocrine cells, well, it shouldn't be surprising that they're going to secrete chemical messengers, some of which have to get into the bloodstream. And if they get into the bloodstream, well, that makes them hormones, right? So, I want you to make a note about the hormones as well as those that act as paracrines. Remember, if it acts as a paracrine, it influences surrounding the tissues in the area, right? Like it's influencing its neighboring tissues. So serotonin and histamine are going to be secreted and they're going to stay here in the stomach where they can influence things. Whereas somatostatin and gastrin will be getting into the bloodstream so that they can act on other organs. Now, we talked about the secretions of the stomach, and I told you that the stomach had some pretty nasty secretions, right? This is a harsh environment. If you think about pH in the body, like what was the pH of the blood? 7.35 to 7.45, so just a little on the basic side, right? Mm-hmm. And that's the way a lot of the body's tissues and fluids are, somewhere around that slightly basic pH. So to have a fluid in the body that has this extremely acidic pH, less than 3, right? Typically. That's a big deal. Not only do we have this high acidity, but we also have enzymes that can break down proteins. So the stomach has to have ways to protect itself from itself. And one of the ways it's going to do that is with a mucosal barrier. Notice what this mucus has in it, bicarbonate. That's something we've talked about before, right? Mm-hmm. What was your equation for bicarbonate? All right, we had CO2 plus water, H2O, and that made what? H2CO3, which is carbonic acid. And then that's not very stable, right? So that dissociates. What does that make? Hydrogen ions, good. And bicarbonate, HCO3 minus. Remember how I told you this keeps coming back up, right? So if you don't learn it, it doesn't go away. This isn't the last time you're going to see it. We also talked about the fact that this is a reversible reaction, right? So it doesn't always have to make bicarbonate. Sometimes it can run the other way and make more carbon dioxide. What regulates that? The direction that it goes?
Where we have less.
Well, the lungs and the kidneys.
Well, those are the organs that are going to help to modify that, but I'm just talking about the reaction itself.
The high and low, wherever we have concentration.
It has to do with concentration. Remember how I told you that the two sides of this equation should be in equilibrium. They should balance each other. So if one side becomes too high, that's going to cause the reaction to run the other way. Well, if this mucus that's lining the inside of the stomach has bicarbonate, how does this reaction need to run then? If I'm spending time talking about this, it's important. I hope you guys are paying attention. Yeah, it should be running backward toward carbon dioxide. And notice what that's going to do. That's getting rid of hydrogen ions. And if we get rid of hydrogen ions, what does that do to pH? It's going to bring the pH back up toward the more basic range. The tricky part when you think about this is that doesn't mean that the mucus is changing the pH on the whole inside of the stomach. If, say, this is the stomach tissue right here, and we've got the lumen up here. The lumen is mostly filled with that nasty pH because of the hydrochloric acid. But it's like we have this very thin layer of mucus that sits right next to the stomach tissues to protect those tissues from the acidity. Do you guys have questions about any of this stuff? All right, then we'll keep moving along. Another way that the stomach protects itself from itself is we find more tight junctions between the epithelial cells. This is important because it ensures that the hydrochloric acid and the pepsin doesn't get into the body tissues where they can start to do things like breaking down those protein fibers that we find in our connective tissues. And you should make a note that the epithelium here is highly mitotic. In other words, it has a very high cell division rate. That way, if one of the cells is damaged, we get a replacement for it almost immediately because we have to maintain that barrier. So, what processes do we see happening in the stomach? We have physical digestion, which, who is responsible for that again? The oblique layer, or sublayer. The oblique sublayer of muscularis externa. We don't just have physical or mechanical digestion, though. We also have what? Chemical digestion. Thank you. And that's because of the acidity as well as because of the enzyme pepsin. Shouldn't be surprising that we also have secretions that help with absorption. So it has secretions that assist absorption. And we know that it acts with helping with propulsion because everything does. But notice I'm giving you a new piece of information here. We change the name of the foodstuff when it's ready to leave the stomach. At that point, it is chyme because it's not just saliva mixed with the food anymore. Now we have hydrochloric acid and new enzymes that have been added to it. So we have to change its name to chyme. Also, do make a note. I don't have it specifically on the slide, but I have told you that it has some absorption, so limited absorption, because we do see water and electrolytes or ions and drug metabolites or medications absorbed here. Yeah.
This it happens always because if you are not taking any drugs.
Well then, if you haven't taken any medications, then it doesn't have any drug metabolites to absorb, right?
Mm-hmm.
But most things that we bring into the body are going to have some water with them, right?
Mm-hmm.
And even if it's a drier substance like a granola bar, we've added water to that, both in the oral cavity and in the stomach. So the body wants to reclaim as much of that water as it can. All right, because the only place where we really want much water to leave is through the urinary system. All right, I told you we'd talk about a little bit of regulation, so I do want to talk about regulation of gastric secretion, which is kind of related to some of the muscle contraction regulation as well. Yeah, Sarah?
With pepsin, it leaves the cell and then it reacts with HCl.
Pepsinogen.
But it leaves, it's outside the cell when it reacts.
Exactly. Yeah, that's what, here, I'll go back. That's what one of these pictures was showing us. So, notice they're showing us, I know we focused on the box up here, right? But notice it's showing us that the pepsinogen was just released from a chief cell. So if it's released from the chief cell, it's going to be in the lumen of the gastric gland, right? And once it's in that lumen, it can now interact with the hydrochloric acid that came from the parietal cell. Yeah, and that's important. We don't want that enzyme to be active when it's in the body cells, because then it can start to act on those cells, and that's a real problem. It will kill them. You're welcome. All right, so let's look at some regulation. Regulation of gastric secretions is going to happen in 3 major phases. What does cephalic mean? It's a region of the body. Where is it? Yeah, it's the head. It's the head, not just the brain. It's the head. The cephalic phase, the reason why they call it cephalic is because stuff that the head is sensing is going to regulate it. So like if we see food or smell food, that's going to influence gastric secretions. Like I told you last time, before the organs ever see the food stuff, they start to prepare themselves for it.
Because sensorial experience, right?
Exactly. The next major phase is the gastric phase, where the food is actually in the stomach. So we're going to see how that influences stomach secretions. And then we have the intestinal phase. As the chyme enters the small intestine, that's going to influence the stomach. And at first, that's going to have a very brief stimulatory effect. But very quickly, that's going to become an inhibitory effect where the small intestine is sending signals to the stomach, telling the stomach to, hey, slow down. And that point should kind of make some sense, because what's one of the processes that happens in the small intestine? Well, emulsification does, that's true. And that's where the rest of our breakdown happens, right? Our chemical and mechanical breakdown. But what else happens there? Absorption of our nutrients. So we've got to be, one of the reasons why we have to be careful about regulating how quickly stuff gets into the small intestine is because, well, think about it. If we shove the chyme too quickly from the stomach into the small intestine, that's going to force it through the small intestine too quickly. And if we force it through the small intestine too quickly, well, now the small intestine doesn't have time to finish breaking it down so that it can absorb stuff. Which means that all of the work, all of the other organs have done before it is kind of a waste of energy if we're not getting all of the nutrients that we need to out of the food. I know that this chart looks a little intimidating. I'm not going to make you memorize every single point here, okay? But we are going to look at some specific things. So first off, notice they're showing us stimulatory events on the left, inhibitory on the right. Which end of the nervous system is going to help us with those stimulatory events? Parasympathetic, exactly. And who for the inhibitory side? Sympathetic, exactly. Also notice that they've color-coded the chart for us, so the upper part with kind of the yellowish-brown background is related to things in the cephalic phase. Green is showing things that are happening in the gastric phase. Gastric means stomach. So anytime you see gastric, that means stomach. That's why so many of our terms had gastric in the name. And in red, we've got the intestinal phase. So notice in the cephalic phase, we're going to focus on the stimulatory side first. It's just like I said, right? If we see food or smell food or taste or even think about food, that's going to help us to be hungry. That's also going to help to cause signals that are happening through the vagus nerve, which we know is where our parasympathetic output comes from, to stimulate secretions in the stomach. That's why this is a solid arrow here, because that's showing us stimulation. Once the food enters the stomach, well, that's going to activate different types of receptors. We know we have the mechanoreceptors there. They're sensing the stretch. We have chemoreceptors that are sensing what kind of food stuff we've gotten into the stomach. They're also sensing pH. And so we can see that stomach distension, activating stretch or mechanoreceptors, food chemicals, activating chemoreceptors, leads to an increase in stomach secretions, because once again, all of these arrows are solid lines. In the intestinal phase, the chyme has begun to enter the intestine. And so, just like I said, that has a brief stimulatory effect. The inhibitory effect is a little more important here. Okay? Yeah, Michael.
A question about the intestinal phase.
Mm-hmm.
When it says inhibitory effects are at work, so that would be baroreceptors and chemoreceptors, or?
The baroreceptors and chemoreceptors are going to help with the inhibitory effect that I'm emphasizing. That is right. Yeah, and that's partially because, think about it, chemoreceptors help to monitor pH. The chyme was just created because we mixed the bolus with a bunch of hydrochloric acid and that enzyme pepsin, right? So once that stuff that has a low pH enters the small intestine, well, now the small intestine is going to have to deal with that pH to protect itself now from these nasty secretions from the stomach. So that's part of the reason why, that's another part of the reason why the delivery of the chyme is going to be really slow to the small intestine. So since we're talking about that, notice they're showing us that as the duodenum fills with chyme, and also as our chemoreceptors sense the acidity of the chyme, that leads to reflexes that have an inhibitory effect because the arrow is a dotted line now that's showing you inhibition. Within the other phases, notice that emotions can decrease secretions of the stomach. That's why if you're really anxious or sad about something, you won't feel as hungry. But also notice that if the pH becomes too acidic, the stomach is going to regulate itself. The chemoreceptors in the stomach sense that. And so then they inhibit secretions of the stomach because, hey, we don't want the acidity to get too high here. Otherwise, the mucus that's lining the stomach won't be able to handle that pH. We're almost there, guys. Just a couple more points. We know the stomach is going to assist with propulsion. Otherwise, it would never be able to get the chyme into the small intestine. So there will be peristalsis that happens here that is still done by muscularis externa. However, I want you to make a note that as the contractions happen, they kind of start up toward the fundus and work their way through the body toward the pylorus. Once we hit the pylorus, that's where we have the strongest contractions in the stomach. Even though the pylorus has the strongest contractions, it's only going to cause 3 milliliters of chyme to enter the duodenum at a time. That's almost nothing. That's like a couple of squirts. So the rest of the stuff that it's contracting to move is going to be moving backward up into the body in the fundus. And that's important because that's probably bigger chunks of things that we need to digest down more. So that's giving us more time to work on digesting those things. And that's what this diagram is supposed to show us. So notice we're seeing peristalsis happening. Right? That's what those little arrows are showing us. And as we get the chyme down toward, or as we, well, partially we're getting a chyme down toward the pylorus, but also as this wave of contraction hits the pylorus, we get stronger contractions, and that's why they call that a grinding step. Right now, though, during this grinding step, the pyloric valve and sphincter are closed. Once that opens, it's going to open just enough to let a tiny bit of chyme into the duodenum. The rest of the chyme gets moved back up so that we can mechanically digest it and chemically digest it more. And that's why they're calling it retropulsion. Remember, retro means behind. So rather than moving the food stuff toward the end of the tube, we're moving it backward up the other way.
Is it like segmentation?
What?
Is it like segmentation or it has nothing to do?
It's kind of like segmentation, except with segmentation, it is. It's related to segmentation. Yeah, that's the easiest way to think about it. So, this is a point we've already kind of hit, right? As the chyme enters the duodenum, there are going to be receptors in the duodenum that monitor the chyme. As the chemoreceptors sense the low pH, and as the mechanoreceptors sense the stretch as the duodenum gets filled with chyme, they're going to send signals to inhibit the stomach. And that is called an enterogastric reflex. Not all chyme is going to spend the same amount of time in the small intestine. So notice, if the chyme has a lot of carbohydrate, it's going to move through the duodenum pretty quickly, whereas if it's lipid rich, it's going to spend around 6 hours or so in the duodenum. Why does that kind of make sense? Why would carbohydrates move through more quickly?
Easier to break down.
Because it increase the blood sugar levels?
Well, that's what they can do, but think about your digestion. Where did the chemical digestion begin for the carbohydrates? In the oral cavity, exactly. Whereas where does most of the chemical digestion, where does that really begin? In the small intestine for the lipids, right? Because we can't really do much chemical breakdown until emulsification happens, until we break up that big fat globule into the smaller fat droplets so that the enzymes can act on them. That's where I was going to leave us for today. If you have questions about something, just come on up and ask me about that during the break. Otherwise, you guys can go ahead and take your 10 minutes.